All right, now let's get into some more variations on Mendel's laws. There is this case of incomplete dominance. Incomplete dominance means that neither allele is dominant over the other. And instead, when you have a heterozygous genotype, you have a mixture, a blending of the two parental genotypes. Okay, so in this case, we've got one true breeding red flower, big R, crossing with a true breeding white flower, WW. Okay, and when you bring that big R and that big W together, because of incomplete dominance, neither allele being dominant over the other, you get a new phenotype. That's a blending of those two genes. So what happens if you have paint, red paint and white paint, and you mix it together? What do you get? Pink paint. Okay? So then heterozygous flowers, RW, are going to be pink. A blending. Okay? This does not happen incredibly often, but there is this case where you can have this mixture, this incomplete dominance in certain traits. Okay? Now, of course, what happens if that whole first generation is pink, RW, and then you cross the two RW pink flowers together? That's what we've done here. So guess what? Pink flowered male, pink flowered female, and when you fill out the Punnett square, guess what? The two original parental phenotypes are expressed. 25% red, big R, 25% white, big W, and then 50% though are going to still express that mixed blended phenotype because they are heterozygous, RW. Okay? So if I ask you a question that says, if red flowers cross with white flowers and all the offspring are pink, this is incomplete dominance. That's what you should tell me. Incomplete dominance, as opposed to complete dominance. These other things that I've been talking about are complete dominance, right? If you've got a big R and a little r, only the trait in big R is expressed, that's complete dominance. Okay? This is incomplete dominance, which results in a new phenotype. That's a blending of the original parents' phenotypes. Okay, now I do want to spend a good amount of time on talking about blood types, because blood types are really cool, okay? But they can also be a little bit confusing sometimes, okay? So consider this fact, that there are more than 2 alleles in the human population for blood type, right? There's the A allele, there's the B allele, and there is the O allele, 3 alleles. So this is a case of multiple alleles. But each person is only going to carry 2 of these alleles. All right? However, because of the dominance and recessive relationships going on here, we do have 4 possible blood types, right? You can have A blood, B blood, AB blood, or O blood. And I'm going to show you why. But the whole reason why the AB blood type exists is because the A and B alleles are co-dominant to each other, which means that both alleles are expressed. Okay, this is not a new phenotype technically. It means that both alleles are expressed. Okay, and there's going to be a really useful picture on the next slide that illustrates this. All right, so let's talk a lot more about blood types. Okay, this is a very helpful diagram. So the phenotypes, when you have your blood type tested, which do you guys know that one of the best advantages of going and giving blood is that you get to find out your blood type. Not only are you doing a good thing, right, to help out the blood banks, but you also get to find out your blood type for free. Because on its own, if you wanted to find out what blood type you are, it would cost a lot of money. Okay? But if you go and donate blood, you usually find out for free. Okay? And it's a useful thing to know. All right. So, but when you get your blood type tested what they tell you your blood type is your phenotype, okay? So phenotypes for blood type can be O blood, they can be A blood, they can be B blood, they can be AB blood, okay? Now in terms of genotypes, okay, we're going to list out the possible genotypes that can give you these phenotypes, all right? But when considering this, you should remember that the alleles A and B are dominant. They are dominant, and O is recessive. Okay, the O allele is recessive, A's and B's are dominant. Okay? So if you are O blood, what does your genotype have to be? OO. You got to have 2 copies of O. Okay? It can be symbolized using those little undercase I's, but I think it makes it easier to do your Punnett squares just using the O's alone. Okay? It makes it less confusing. So anytime you see an undercase I, that means O. Okay? It means a lack of carbohydrate attached to your blood cells. Okay? See how in the picture there's no carbohydrate on the O blood cell? That's because O blood does not have carbohydrates attached to their blood cells. Yes?
To find this out for the blood cells, would you use the 4 squares or the 16 squares?
Just 4. Just 4. Okay, make it simple. We're not going to do anything else in 16 squares. Okay, everything else will be simple monohybrid crosses. Okay, but there's special rules for blood types, okay? Now, if you are type A blood, you probably do not know if you are homozygous for A blood, AA, or if you are heterozygous for A blood, AO. Okay? So if you're A blood, you could have 2 possible genotypes. You could be homozygous for A or heterozygous for A. Okay? Remember that O is recessive. So if you're AO, you're A, because A is dominant to O. Okay? If you are B blood, it's the same situation. You can be homozygous for B blood, or you could be heterozygous for B blood. And you probably don't know. Okay? If you're AB blood, you're AB, right? Your genotype's got to be AB. The only reason why this blood type exists is because the A and B alleles are equally dominant. They're co-dominant to each other. All right? So take a look at that. This is really important to remember, okay? Because if I ask you a question, well, let's do an example right here, okay? Let's say we're going to cross a man who is heterozygous for A blood, okay? So we got a dad who's heterozygous for A blood. What's his genotype? AO, okay? So half of the sperm carry A, half the sperm carry O. We're going to cross him with a woman who is heterozygous for B blood. So what's her genotype? BO. So half of her eggs are B, half of her eggs O. So let's see what we got here. What are we going to get? AB, BO, AO, and O. Take a look at that. That's awesome. A cross between a parent who is heterozygous for A and heterozygous for B will give you a 25% chance of having babies with any of the blood types. Okay? Before I mentioned this example, I had a student once who really freaked out about that. Okay? She was like, well, but I'm O blood, and my dad is A, and my mom is B, and my sister is AB, so is one of my parents not my parent? Kind of thing. Is my paternity in question? And I was like, I had to think about it for a second. Of course, I don't know for sure, but I was like, this is how it's possible. And I said, well, probably your mom is heterozygous for A, your dad's heterozygous for B, okay? There you are. There's your sister. Yeah, it's possible. I don't know for sure, but yeah, it's possible. 25% chance of any of those blood types popping up in that generation. Okay? O blood. Do you guys know what happens if you get the wrong type of blood? Okay? Your blood will clot, and you can have serious health implications or die. Okay? So take a look, though. One of these blood types is a universal receiver, and one of them is a universal donor. Again, you can tell by what it is based on that diagram right there. It shows you what condition blood would clot. Okay, so who is the universal donor? O blood. Okay, for example, I have type O blood. O blood is the most common blood type amongst human beings. Okay, and O blood is the universal donor. Anybody can receive O blood because you don't have those carbohydrates on your cells, so you don't have the antibodies to fight them. Okay, and so anybody can receive O blood, not a problem. And AB blood is going to be your universal receiver. They can get any type of blood. Okay? Any type of blood people with AB blood can get. And no clotting happens. But really bad situations happen if you're O blood and you get A or B or AB blood, forget it. Okay? Same thing with those couple other examples. Okay. How much time do I have? All right, I'm going to finish up a couple more examples here, and then we'll finish it up the rest on Tuesday. So inherited human disorders. So in some cases, genetic diseases that people have can be because of dominant inheritance, where you only need to inherit one copy of the dominant allele to have a trait. Okay, it could be a harmful trait. And so like the allele that causes dwarfism, okay, so reduced stature, okay, tend to have some abnormalities in spine and skeletal structure. It can lead to a lot of pain. That is a case of dominant inheritance. So let's take a look at, I'm going to go back to this slide, but let's look at this example, okay. So if you have two parents that have dwarfism, but they're heterozygous, big D, little d, big D, little d, because they have the dominant trait, they have the condition, right? But if they have babies, and they're both heterozygous, they do have a 25% chance of having a normal, not affected by dwarfism child, okay? But 3/4 of them are predicted, do have the probability of having dwarfism like their parents. Because all you need is one big D allele to have it. In the case of recessive inheritance, you need 2 recessive alleles to show the disease, to show the trait. Okay? And so if both parents are heterozygous and they carry the allele, they can pass that trait on to the parents, to the offspring. They can pass that trait on to the offspring. Okay? Generally, the probability of recessive alleles does increase with inbreeding and mating between close relatives. Okay, so here's an example of recessive inheritance. Okay, so you can see that if you've got, if we're talking about deafness here, okay, you've got 2 parents who are normal, they can hear, but they both carry the deafness allele. When they have babies, they have a 25% chance of having a child who's deaf. All right. Okay. Here is a look at just some examples of genetic abnormalities. This kind of relates to that assignment that you guys have to do, your other writing assignment. So you can research a different genetic disease, or else you can even pick one of these, okay, and talk about it. But I'm going to kind of leave that discussion for that assignment. All right, so these are just some examples of conditions, whether they're recessive or dominant disorders, what they cause, and then the proportion, the frequency of these occurring in populations. Some of them are more common in different ethnic groups and racial groups than others. Okay. Okay, pleiotropy. Pleiotropy means that you have one gene that influences many characteristics. So the gene that causes sickle cell is an example of this, pleiotropy. Okay? One gene that influences many characteristics. So the gene for sickle cell disease affects the type of hemoglobin you're going to produce. It's going to affect the shape of the red blood cells. Okay? It specifically causes some red blood cells to sickle. And it can it can cause anemia and organ damage, especially if untreated for a long period of time. But it is also related to the susceptibility to malaria. This is pretty cool, Okay. Now, where in the world is malaria, a disease transmitted by mosquitoes, most common? Africa. In any tropical areas of the world, okay? But in Africa, okay? And so what group of humans is there a higher incidence of the sickle cell gene? Amongst people of African descent. Because for people who live in Africa, at least carrying this gene has benefits. Because if you carry this gene, you have resistance to malaria. And malaria is a scourge that wipes out a huge amount of people around the world every year. Okay, it has such a huge negative impact on the human population. Okay, and this gene actually exists due to an adaptation to survive malaria. Okay, so let's take a look at that. Okay, so let's take a look at the breakdown of genes. Sickle cell is from recessive inheritance. So to have sickle cell disease, you have to be homozygous recessive, little s, okay? Sickle cell. But the other genotypes that are possible, right, are big S, and also what? Big S, little s, okay? Now, if you are big S, you don't have sickle cell disease, you're normal, but if you get malaria, you're probably going to die of malaria. Okay? Now, the person who is little s is going to be negatively affected by sickle cell disease. Okay? However, they are going to be almost immune to malaria. Okay? The best thing to be is heterozygous, because with this, you are still considered normal in terms of your cells, not sickling, and you are resistant to malaria. So over generations, amongst people who live in malaria-plagued regions of the world, this genotype has become the most persistent and abundant, because these are the people who tend to have the most babies and survive longer and pass this gene on to the next generation. It's called a heterozygote advantage. You're a carrier of the recessive allele. So you have that resistance to malaria, but you do not have the negative impacts of sickle cell disease. Yes?
So the SS, the third one, are those both recessive?
These are both recessive. Little s, with thus the line underneath. Okay? All right, guys, I guess I will end it right there. I only have really one major topic left to cover, which is sex linkage. Okay? So thank you for your attention, and I will see you guys on Tuesday. Where I left off last Thursday was on the topic of pleiotropy. Remember that? The fact that one gene can influence many traits. And so one of the examples was the gene that causes sickle cell disease, but that's also related to a person's susceptibility to malaria. Okay? One thing that I wanted to mention before I wrap that up is that the incidence of heterozygosity of the sickle cell gene in the African American population used to be 1 in 10. Okay? 1 in 10. What does that mean, heterozygosity? If someone is heterozygous for the sickle cell gene, what is their genotype? And we use S's for that. What is the genotype of a heterozygous condition for sickle cell? Well, using S's? Big S, little s. Okay? So in African Americans in the United States, the incidence of heterozygosity for the sickle cell gene used to be 1 in 10. Okay, that's where it started. But then over time, since malaria is not being a selective force in the population here in North America, the ratio is actually dropping. It's now 1 in 12. Okay, so the odds of having people who are carriers of the sickle cell disease is actually declining. And so now it's not 1 in 10, it's 1 in 12. Okay, because malaria is no longer a selective force that's keeping this allele in the population. Okay, so it's interesting. Anybody have any questions on that? Okay. Another thing that I just want to mention very briefly is the concept of polygenic inheritance. Okay, in most cases I've discussed, one gene to control one trait, right? Or in the case of maybe a dihybrid crosses where we're talking about 2 traits and tracking the inheritance of those 2 traits right at once. The concept of polygenic inheritance is a bit more complicated. This is a situation where many genes influence one trait. It could be 3 separate genes all together influencing one trait, like eye color or skin color. And that's, in fact, how it is. So an example of the genotype for eye color or skin color would look something like this. Okay? So you've got 3 separate genes influencing that one trait, the A gene, the B gene, and the C gene. All right? Now, to predict probabilities of inheritance for eye color or skin color, being that we have these complex genotypes, guess what kind of a Punnett square you'd have to make to determine probabilities of inheritance between 2 individuals with this genotype? Okay, so the dihybrid one is a 16 square Punnett square box. You know how many boxes you would need for this? 64. Am I going to ask you to solve problems using a 64 square Punnett square box? No. Okay. Don't worry about it. However, if you do want to practice it, there is a really cool link that I have to your D2L page. It's called Genetics Practice Problems. Okay? And there's all sorts of practice problems that range from simple monohybrid crosses to dihybrid crosses, okay, to ones like this where you can calculate the probability of inheritance with skin color using a 64 square Punnett square box. Okay? Trihybrid crosses. So you might want to check that out if you really want to practice your skills. Okay, but that's why there's such a range in skin color tones and eye color, right? Think about all the eye colors, right? There's black, there's brown, there's hazel, there's green, there's blue, and there's different intermediates between that. Okay, that's why, because it's a case of polygenic inheritance. Okay, now let's look at sex linkage. This is the last type of a problem that you will have to be able to solve, is sex linkage problems. And those of you who are in lab class already know from yesterday a little bit about this, okay? But those of you who are in lab tomorrow or later on today, you'll get some practice with that. But as an introduction behind that concept, we have to review the sex chromosomes, right? So the sex chromosomes are X and Y. They are not homologous. They're not the same size, right? You can see that the X chromosome is huge compared to the Y chromosome. All right? And males are XY. They get one of each chromosome. They get the X chromosome from their mom, and they get the Y chromosome from their dad, and that's what made them a male. Okay? Females, in contrast, are XX. One X from mom, one X from dad. Now, some genes are linked to specific chromosomes. Those genes that are either linked to an X chromosome or to a Y chromosome are called sex-linked genes. And so X-linked genes, genes that are linked to the X chromosome, and there are many, okay? And that's because the X chromosome is huge. And there is so many additional traits and characteristics that are linked to that X chromosome. Okay? In contrast, the Y chromosome, you'll notice, is very, very small. There's not much extra stuff linked to the Y chromosome. Mostly the Y chromosome codes for maleness. Okay? But X-linked genes can be passed from mother to son, okay, or mother to daughter. X-linked genes can also be passed from father to daughter. And Y-linked genes, though, have to be passed from father to son. They cannot pass to daughters. Okay? So let's take a look at some examples of sex-linked genes and their ramifications. Okay. A lot of disorders, recessive sex-linked disorders, tend to be linked to the X chromosome. And the reason is, like I mentioned before, the X chromosome is huge compared to the Y. Okay? I don't know if you guys can see this poster that well over here, but it has all the different chromosomes that we have in the human genome that we get from each sperm and egg when fertilization occurs. And it has a listing of all the different traits that are coded by each chromosome. Can you see this? This is the whole list for the X chromosome. This is the Y. Okay? So you can see that there's a whole bunch of stuff coded for in the X chromosome, and very, very little other than maleness in the Y. Okay? So there's a lot of extra stuff linked to these X chromosomes, and some of them are harmful conditions. Okay? One of them is hemophilia, a recessive X-linked condition. What does hemophilia cause? You guys don't have any idea? Somebody has to know it there. Don't be shy. What? Something about blood, right? Yeah. The inability for blood to clot is the problem. And so basically people who are hemophiliacs, if they get cut, a serious cut, they can actually bleed to death from that because of the lack of blood clotting. Okay, you know how if you get a cut, you know, you put pressure on and it stops? People who who have hemophilia, that doesn't happen. Okay, they need some extra special medical attention. So hemophilia is an X-linked disorder, recessive condition. Color blindness also, and Duchenne muscular dystrophy. Now all three of these examples of X-linked conditions are more common in males than females. And this is the reason, because males only inherit one X chromosome, right? From their mom. And if the recessive allele is linked to that X chromosome, they've got the condition. Females can have these conditions too, but they'd have to inherit two X-linked chromosomes, two X chromosomes that both have that recessive allele linked to the X chromosome. Okay? So for example, if we're talking about hemophilia, this would be the genotype of a male with hemophilia. Okay, undercase allele, recessive, remember that? This male would have hemophilia. In order for a female to have hemophilia, she'd have to have inherited this. The X chromosome with the sex-linked recessive allele for hemophilia from both mom and dad. And that's very unlikely. It's very uncommon, which is why these conditions are much more common in males than females, because males only need to inherit one affected X chromosome to have the trait. Okay? Now, let's look at this pedigree here. This is a pedigree, this sequence of circles and squares that are all kind of connected. It's like a family tree. All right? And pedigrees are used to trace the inheritance of a trait across generations. This is especially useful when you have genetic conditions that you want to be able to track in your family. So if you have a half-filled circle or square, that means that somebody is a carrier of the trait. And if they're a carrier, what's their genotype? Are they homozygous or heterozygous? Heterozygous. Carriers are heterozygous. Okay? And if it's a white circle or square, that just means they don't have it at all. They're not carriers and they're not affected. And if you have a completely closed circle or square, that is a person in the family who has that recessive X-linked condition. Okay? So let's take a look at this pedigree right here. Okay, who do we start with? Who is Queen Victoria and Albert? Where were they the royal family from? History, guys, come on. What do you think? Guess, you know, royal families, what country still has royal families and stuff? England, right? Yeah. They were the Queen Victoria was the Queen of England, and her husband was Albert, her first cousin. Okay? Yeah. Now, they had lots of kids, a whole boatload of kids. Okay? We're going to follow the pedigree, though, to their daughter Alice, who, just like her mother, Queen Victoria, was also a carrier of a recessive X-linked condition, hemophilia. Therefore, what was her genotype, do you think? What do you think both Queen Victoria and Alice's genotype? Okay? Okay, what kind of X? What kind of H? So X what? X what? She's a carrier, so heterozygous, right? So X, what kind of H? Not small. Big H, and then this X is what? Little h. Okay? This is the genotype for this condition of both Queen Victoria and Alice. Okay? They are carriers, so they are heterozygous for the condition. They don't have hemophilia themselves, but they carry it. Now, Alice and Louis, they had a daughter, Alexandra, who, just like her mother and grandmother, also was a carrier of hemophilia. Now, she married the Tsar of Russia, Nicholas II. And this is them and their family right here. So this is Alexandra, that's Tsar Nicholas of Russia. And in fact, this is the last royal family of Russia. Okay? Anyone know what the name of this family is? Like what their last name is? That's probably a tougher question. This is where the whole legend of like Rasputin and all that comes in. And Anastasia. Okay? Anastasia, in fact, is right here. Okay? So, anybody know yet? The Romanovs. Okay, the Romanov family. This is the Romanov family, the last royal family of Russia. And in fact, they were all murdered right after this picture was taken. Okay, after the communist takeover of Russia and it becoming the Soviet Union for that period of time that happened. Okay, so this is a really famous picture and it's a really famous case. Okay, the whole thing with Rasputin came about because Queen Alexandra had hired Rasputin. He was kind of like a magician, sort of a mystic man from the Ural Mountains. And it was believed that he basically told the royal family that he can cure their son, Alexei, who has this horrible condition where he just bleeds and it doesn't stop. Okay? Because after all, Alexei, he was the only male of the family and he was expected to inherit the crown and be the king. Okay? But he was very weakly because he had this condition, this genetic condition. Of course, there was nothing that Rasputin could do to cure him of this condition because it was in his genes, it was genetic. Okay? But that's sort of a whole story unto itself, is the influence of Rasputin on this family. But as a whole, okay, we can see that Alexandra then passed on the allele for hemophilia on her X chromosome to Alexei. Okay? And that's why he has that condition of hemophilia. And so Alexei's genotype, therefore, is what? X, little h, y. Okay? Now let's see why this happened. Okay? So we're going to do a little Punnett square. And we're going to cross Alexandra's genotype, X, big H, X, little h, with Tsar Nicholas, who was normal. He was not a carrier of hemophilia. And so if he's not a carrier of hemophilia, what's his genotype? What kind of H on his X? big H, Y. Okay, so this is the cross we're going to do. So we just have to do a simple monohybrid cross, a 4-square Punnett square box. Okay, let's start off with mom's genotype. Okay, so when she made eggs, half of her eggs were what? X, big H, and the other half of her eggs were X, little h. Okay, Tsar Nicholas, when he made sperm, half of his sperm were X, big H, and the other half of his sperm were Y. Okay, so when they had kids, they had a 25% chance of having a normal non-carrier daughter, and they had lots of daughters, okay, 4 of them. They had a 25% chance of having a normal but carrier daughter, okay. They had a 25% chance of having a normal son, and they had a 25% chance of having a son with hemophilia. And this is Alexei right here. Okay, so that's how you would do a sex linkage problem. You would set up the parental genotypes, and you would set up your Punnett square and fill in the box to predict the probability of inheriting a sex-linked condition. Okay? All right, well, that is the conclusion for the Chapter 9 lecture.